Folks, morning. I see we have 8, so let's begin. We're going to run through chi-square test today, as well as finish up genetics and move on to mutation. Can I assume that you've all done chi-square test already? Wait, have you done resource methods or statistics yet? You should have, right? Resource methods, statistics. Have you done either one of those yet?
No, sir.
Or is it next semester for that? Oh, but they should be in 2 separate semesters. You're not doing research methods right now? That's strange. Okay. Um, well, we're going to cover chi-square test today, which is one of the basic tests you're expected to know to do in statistics anyway. So that and the studentized t-test, which you will probably do in research methods. So I guess getting a jump start on it is not a bad thing, right? All right, so you should be seeing my screen. If not, you can let me know, please. All right, so the last thing we did was epistasis, right? I want to do, and we did different types of epistasis. So we spoke about um, pathway epistasis, duplicate gene epistasis, and I want to give one more example of epistasis, and you can tell me which one of these types of epistasis this is, right? So let's just make sure it goes again. So we have epistasis, right? And we look at 3 types. We have dependent gene epistasis, right? Where one gene directly controls the expression of another gene. We have enzyme pathway epistasis, in which one gene controls an enzyme that is required for another gene to function properly. We have duplicate gene epistasis, which you have multiple genes both affecting the same gene in the same way. Okay? So those are our 3 types that we looked at. So let's now look at Labrador retrievers. You can tell me which one of those 3 this one will fall into, okay? So Labradors, as I'm sure everybody knows, are one of the most popular breeds of dog in America, right? Funny enough, I don't remember seeing any in Jamaica. I mean, chances are there must be Labradors here somewhere, right? But I just don't remember seeing any. That said, we have a case here where we have the black color being dominant and the brown color being recessive, okay? So we start there. So simple monohybrid cross gives us this. Of course, as we know, this is of course the F1 generation, assuming that the P generation, of course, was this. Right? So far so good. But instead of getting brown all the time or black, sometimes you get a yellow lab. Right? Now, as it says that this could be incomplete or codominance that they're asking, right? But it says there's another set of alleles that work with your dominant and recessive color. And this allele is allele E. And allele E determines whether or not the black or the brown color shows up in the coat. Right? So because of that, when we have a functional allele E, then the animal can be black or brown. So this, for example, here, Well, all right, let's do what we normally do. So we'll do this, all right, which is based on, of course, that. So remember now, the first thing we do is we determine the gametes that you can get, all combinations of B and E. So capital B, capital E, capital B, common E, common B, capital E, and common B, common E, which of course how we combine these, right? And of course, since it's two of the same parents, they get the same thing on the other side. Now, remember now that common B's give us brown, right? Capital B gives us black. And of course, common E gives us yellow, all right? And that said, we should be able to determine the colors of all of these dogs here, okay? So for example, we know that the first dog here would have to have a black coat because capital B, remember, equals black. The next dog here also has a black coat, and so does that one, and so does that one. Nice and easy, right? So does that. Now, this one, of course, will give us a yellow coat because once you have the recessives for E, you get yellow. And that one, of course, is black again. And that's another yellow. And that's black. And black. This one is finally a brown. And that one is also a brown. That's another black. And that's a yellow. And the brown. And the last one, of course, is a yellow. Okay? So far, everything I've done, you've seen me do it before. So you should understand how I come by these. Then we count up. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 black. And then 1, 2, 3, 4 yellow. And 3 brown. Right? So a 9-4-3 ratio or 9-3-4. Either way it is, right? So based on what I just said, can you please tell me what sort of epistasis this is? Is it dependent gene epistasis? Is it enzyme pathway? Or is it duplicate gene? Julian, what do you think? Julian Edwards.
Yes, sir. Mm,
Okay, still waiting on a response.
sir, I'm not so sure.
Okay. Kalicia, what do you think? Kalicia Swaby. Kalicia Swaby, are you there? All right, so what type of epistasis is this? Can so please type it in the chat. Can I repeat the question? Come on, people, it's 8 o'clock in the morning. It's like I'm talking to myself. What type of epistasis am I seeing from the dogs. That's all I've asked. We're 10 minutes into the thing and people don't know what's going on. Sarah Ann McIntosh, are you there?
Yes, sir, I'm here.
What kind of epistasis is this?
Sir, could you go back? Not there. No, right there.
Okay, and your responses? Hello? Anybody there?
Sir, it's a dependent gene.
Finally, an answer. Thank you very much. Ruth and Douglas, what kind of epistasis is this?
Dependent gene epistasis.
Okay, that's 2 for dependent gene. I'll take one more. Tasania, what kind of epistasis is this? Tasania, you just wrote gene. Many of them have the word gene in it. You have duplicate gene, you have dependent gene. I don't know which one it is, so you have to let me know, please. Tasania, I'm still waiting. You just wrote gene. I need to know which one without the genes. Is it dependent gene or is it duplicate gene? Okay, duplicate. Okay. 3 people say duplicate gene. Okay, fine. The answer, of course, is duplicate gene epistasis, okay? You need to be able to quickly parse the difference between the 3. Okay? All right, moving on. Now, it mentions, of course, that your normal dihybrid ratio, if we're looking at a Mendelian cross, would give us 9331. Okay? But of course, when we do the dependent gene, we tend to get a 934 cross instead. Okay? Now, let's also talk about lethal genes. Lethal genes are genes that when they are in ascendance, they cause the death of the animal. Most lethal genes tend to be recessive, okay? So because they tend to be recessive, you require 2 of them in order for them to have their greatest negative impact and therefore end up killing you, okay? So for example, cystic fibrosis. I think I've mentioned cystic fibrosis already, but just in case, let me mention it again. Cystic fibrosis is caused by a CFR gene, all right? This, I think I also mentioned too, is the most common genetic defect amongst Caucasians, right? Once again, it is a recessive gene. And what happens with cystic fibrosis gene is that it causes the production of, well, it affects chloride ion channels in the person's body, right? All right, let me just put it up real quick as an example. Right? So because it affects the CFR gene, it leads now to chloride channels in the cells not working properly. And I won't get too deeply into how that causes the issue. Just be aware that chloride ions, when that channel doesn't work properly, the mucus that is produced by the person's body is extremely thick and sticky. And because of that, that mucus is difficult to move. So there's a tendency to block up the tubes and passages, and even when it comes to joints, anything where you have mucus being put, it negatively impacts that area of the body, right? Particularly in the lungs. So it's, over time, cystic fibrosis will cause the lung, the bronchioles and the alveoli in the lungs to fill with mucus, which they cannot remove because of how thick it is, and that eventually leads to the person's death, right? So it is not immediately fatal, but fundamentally, once you have cystic fibrosis, even with treatment today, right? Even with the best treatment in the world, it's still going to shorten your life. And there's not much you can do about that. You can live a little longer with treatment, but you're still going to die, okay? Another example of a lethal allele can be considered sickle cell anemia. Because once again, if you have 2 alleles for sickle cell trait, it is going to shorten your life. There's no way to get around it, okay? Nobody lives it to be their 80s with sickle cell, with full-blown sickle cell, okay? Without treatment, most people with full-blown sickle cell will die before they hit their teen age, right? So lethal alleles, once they show up, you go away. Simply put. Most of them are recessive, right? And remember, they do not have to be immediately fatal. They simply have to lead to your death. So once again, cystic fibrosis, sickle cell anemia are lethal alleles, okay? That said, of course, as you know, with sickle cell anemia, if there's malaria around, having one gene for sickle cell is beneficial, okay? All right. That said, let's move on, right? So, cystic fibrosis, lethal gene. Next thing we want to talk about is pleiotropy. A pleiotropic gene is a gene that has effects on multiple genes in the body and the way those genes are expressed, okay? So one pleiotropic gene will have a multiplicity of different impacts on the body. So as this is a gene that affects more than one phenotype, it's a pleiotropic gene, right? I think I gave some more information about pleiotropic genes. Where is it? Here. Wait, we already went through this, didn't we? Yes, we did. So we gave the examples of sickle cell as a pleiotropic gene. So many things sickle cell come underneath. Sickle cell is codominant. Sickle cell is pleiotropic. Sickle cell is a lethal allele. There's so many different things sickle cell can be used as an example for, right? All right. I'm assuming you've all you've all gone through the process of learning how to use a pedigree chart, right? Can I assume that? Or do I need to go through it? Y'all let me know, please.
No,
Sir,
sir.
can you go through it, please?
All right, no problem. The basics of a
Sir, can
pedigree
you
chart.
repeat?
Yeah, man, I'm going through it right now. The basics of a pedigree chart, all right, let me make it a little bigger, is that it's supposed to show you how a disease passes from one generation to the next, right? So it gives you an idea as to how that disease is inherited. right? But it can be used for more than just disease, okay? It can be used for any trait. So basically, when you're looking at a pedigree chart, you're looking at multiple generations of a family, okay? And you're tracking whatever phenotype it is through the generations, okay? In this case here, they're tracking widow's peak. The widow's peak, of course, is this right here. When a little bit of hair comes down on the forehead like that, that's a widow's peak. Why it's called widow's peak, I have no idea. And of course, this is what you'd consider to be a normal hairline, okay? Now, based on what we see here, widow's peak is actually a dominant trait, okay? Generally speaking, when it comes to these pedigree charts, what we do is this. The squares here represent males, okay? So if you see a square, it's a male. You see a circle, it's a female. You won't see triangles or octagons on pedigree charts. You only see squares and circles, because what we have in this world are males and females, right? Now, if we have a male that has the disease or the condition you're examining, we normally take the square and we color it one solid color, okay? So let's say, for example, I was doing, you know what? Let's do hemophilia real quick. Let's put together a pedigree chart right now. Why not? To make sure that we're all on the same page. Let's say we're doing a pedigree chart for hemophilia, okay? Hemophilia is carried on the X chromosome, okay? So we can have this kind of situation here. So of course, this male is normal. This male is a hemophiliac, right? But females, we can have a female that's normal or a female that's a hemophiliac. But females can also be carriers of the hemophiliac gene without being a hemophiliac, right? So when you have somebody on a pedigree chart that is a carrier, they don't necessarily have the disease condition, or they're heterozygous for the disease condition, you normally have this circle with banded lines running through it like that, okay? And that person now is a carrier. So instead of hemophilia, you could do once again sickle cell anemia or any other disease like that, right? So based on what we have here in our key, we can therefore do a simple pedigree chart, okay? We can have a female like this, male like that, okay? And let us say the female is a carrier for hemophilia and the male is normal. You then draw a line between them indicating that they've done the deed, and then you draw a line down showing how many children they have. Okay, so let's say they had 3 kids here. Okay, and they have a girl here, a boy here, and another boy here. Okay, now let's say each one of these children reproduced. So the girl here, who is not a hemophiliac, is going to have a child with a male who is not a hemophiliac, right? And they have 2 children, and both their children are normal. All is well. On the other side here, we can have this male, and let's say this male is a hemophiliac. He's going to have relations with a female who is not a hemophiliac, and they too will have 2 children, okay? And they have a daughter, and the daughter is a carrier, and they have a son. Son is normal. On the other side, you have a son here, and his wife is a carrier for hemophilia because it's his cousin, let's say. You know, let's assume these guys are from Pakistan. They love the cousin marriages there. And we do the same thing here. They have 2 kids, and the male is a hemophiliac, right? And the daughter is not a hemophiliac. Okay, so right here on this very small pedigree chart, we have 1, 2, 3 generations of people. Okay, now just based on what I have on this chart here, I can work out which gene is the hemophiliac trait is on. Okay, now I know that the hemophiliac trait is on the X chromosome, but I can prove it based on this pedigree chart. Because if, one second, yeah. If we didn't know the mother was a hemophiliac, right? Let's say we erase this. Let's say the mother is an unknown. Let's just put a question mark above her because this is what you normally see. So we don't know what the mother is. We know the father is normal. We know they have 3 children. We know the female child is perfectly normal. We know one of the male children is normal. And we know one of the male children is a hemophiliac. I can work out what the mother is. How is that? Because a hemophiliac male is going to have an X chromosome for hemophilia and a Y, right? Which means the hemophiliac male can only pass on his hemophiliac trait to his daughter, right? And if you look here, this daughter doesn't have hemophilia or even the trait, right? But you can say, okay, that's one daughter, but they have 2 sons. But guess what? The male cannot pass on hemophilia to his son because the father is going to give the son his Y chromosome, not his X, right? And since one of the sons is not a hemophiliac and one of the sons is a hemophiliac, it means that hemophilia can't be passed on the Y chromosome because both sons have the same Y, right? But one of them has hemophilia and the other one doesn't. So therefore, the mother has to be the one that is passing the hemophilia to the sons, okay? But because one has it and one doesn't, means that the mother must have one normal X chromosome, which she can give to this son here, as well as one with the hemophiliac trait that she gives to this son here. Therefore, the mother has to be a carrier of hemophilia, right? So based on this, you should be able to work out what the genotype of whatever person there is on the chart, assuming you know some of the phenotypes. All right, does that make sense? Yes, no? Taylor's make any sense to you?
Makes sense, sir.
All right. So when you look at a pedigree chart, remember now, squares are males, circles are females. Fully colored squares have the condition you're examining. Fully colored shapes have your condition you're examining. If you see any kind of banding or striping in any of the shapes, it means that they're carrying the gene. They're not necessarily expressing it, right? So based on that, we can now come to this. This is looking at widow's peak and attached earlobes, okay? And this one gives us the genotype at the bottom, which of course we don't really need, because even if we ignore these here, we can work out what's going on, right? So if we didn't know what the parents were, and we see here that you have both males and females have widow's peaks, what does this tell you? Well, it tells me that it can be the Y chromosome widow's peak is on, right? Because males and females have it. It probably won't be the X chromosome either, because the mother didn't have it, the father did, and one of his daughters has it, right? Which means this condition only requires one chromosome to show up, which means it is a dominant condition, okay? So the father passes on his widow's peak gene to his daughter. The mother passes on her normal gene, since she doesn't have a widow's peak, to her daughter, but the daughter has a widow's peak, therefore widow's peak must be dominant, right? You have two sons here, neither of them have the widow's peak. You have another son here with it, okay? Now, when the son marries a woman with a widow's peak, and they have a child, two children, two girls, one comes with a widow's peak, one doesn't, which means that the no widow's peak reappears in the third generation, which tells me for sure, therefore, that widow's peak is dominant and normal is recessive. Because remember, the recessive one can skip a generation and show up later, okay? So even if we didn't see any of these, you should be able to work that out for yourself, right? Same thing with the earlobes here. You have two parents over here that start out with, well, let's see what we have here. It starts out with free earlobes, it looks like, right? So this is a free earlobe. I'm assuming y'all know when the earlobe hangs off of the side of the head, that's free. When it curves down and attaches to the side of the face, that of course is your attached earlobe, right? So two people with free earlobes are giving birth to children with attached earlobes. How does this happen? The free earlobe must be dominant and the attached one must be recessive. Okay? Once again, by seeing how the traits change from one generation to the next, you can work out if they're dominant and recessive, and you can work out which one is dominant over which one, right? So pedigree charts are good for that kind of information, okay? All right. Now, these are some examples of recessive genes that cause disorders, okay? We mentioned cystic fibrosis multiple times already, right? As I said to you, once again, so you should see Cl minus here. The problem is the chloride channel messes up. That leads to the thick mucus, leads to problems. Another example of a recessive disorder is Tay-Sachs disease. And this causes a dysfunction of an enzyme that is, as it says, fails to break up brain lipids. Basically, you get fatty brain disease. And that causes the neurons to start degenerating. And it also causes seizures, blindness, mental problems, of course, because it's affecting the neurons in the brain, and eventually it leads to death, right? So once the symptoms start to show up, usually you're dead in less than a year. You find this in 1 in 3,600 Jews, okay? Cystic fibrosis, 1 in 2,500 Europeans. And as it mentions here, once symptoms start to show up, you normally die within 5 years, right? So once again, a lethal allele does not have to kill you immediately. It can kill you over time. Some lethal alleles will prevent the fetus from developing at all, and the fetus will simply die, okay? But those lethal alleles are less common. Now, let me repeat what I just said. Some lethal alleles show up later in life, right? Late teens, 20s, 30s, even 40s sometimes, before you start to get the effects. But eventually they will cause you to die. While other lethal alleles will prevent the fetus from being viable. So the person dies in utero. So the mother has a miscarriage. Right? Please pay attention to what I'm about to ask you. Why are conditions like cystic fibrosis and Tay-Sachs disease more common than the ones that cause the fetus to die in utero? Why are lethal alleles, these are recessive lethal alleles that kill you later in life, more common in the population than lethal alleles that kill the baby before it's born? All right? I'm going to give you a moment, and then I'm going to ask people that question. It's not a trick question. I'm hoping the answer is obvious too, okay? All right. So let me ask now. Shakeen, what is your opinion?
I think it might have something like some traits not being shown until like later in life. So those traits
Remember,
aren't it?
Tay-Sachs disease and cystic fibrosis are like that. They don't show up until later in life. So what I'm asking is, why is it these diseases are more common than the ones that simply kill you before the baby is born? So lethal alleles that just kill you are less common than cystic fibrosis alleles and Tay-Sachs disease allele. That's what I'm trying to find out. What is your opinion?
Does it have something with the mother's immunity or resistance?
Not really. Remember now, what I'm asking is, why is it they don't show up in a population of people as often? Right? Why aren't there more people with heterozygotes for these lethal alleles floating around in the population? Nothing comes to mind? All right, well, let's try somebody else then. Daniel, you have any idea?
Sir, repeat the question. Right.
Why are lethal alleles that kill you later on in life, normally in your 20s and beyond, more commonly found in a population than lethal alleles that kill you before you're born?
Would it be because if they kill you before you're born, then there would be less to repopulate?
I think I see where you're going with this one. All right. See if this is what you're trying to say. Are you saying that if it kills you before you're born, you can't add to the number of people with the allele in the population?
Yes, sir.
That is correct. Okay. Then add to that the fact. So if you die before you add yourself to the population, then of course your allele dies with you. It can't be counted. Then add to this the idea that if you have a recessive condition like cystic fibrosis, like Tay-Sachs disease, and even like sickle cell disease here, you have a chance to reproduce and pass on your bad gene to the next generation before it kills you, right? So diseases that are based on recessive lethal alleles that kill you later on in life are very hard to control. They're very hard to remove from the population. Whereas if you have a lethal allele that kills you before you're born, it's much easier for you to control alleles like that, right? Because they'll be simply put, there are fewer people available to pass on the allele because some of them die immediately, right? Okay, that said, inbreeding. Inbreeding is a real problem when it comes to lethal alleles and recessive conditions in general, okay? Let's take the UK. United Kingdom, okay? If I say to you, right, that in the United Kingdom, the chances of having a child being born with birth defects is something like 1 in 2,500, right? For the, let's call them the local population, white people, okay? So in the UK, 1 in 2,500 of the white residents potentially will be born with a genetic condition, okay? In the same UK, so this is UK white population. I don't like to say white, but hey, it's basically it is UK white population, right? In the UK, if you are from Pakistani descent, the chance of having a child with developmental problems is close to 1 in 100 and I think it was 130, right? So we have a Pakistani population living in the UK that has like a 1 in 130 chance of having a child with a birth defect. And we have the normal, and I mean, I say normal white population because realistically, they are from there, so we have to consider them to be the normal population, right? The normal population or the local population of white people has a 1 in 2,500 chance, right? If I say to you that in Pakistan, cousin marriages are very popular, can you all see where the link is between the cousin marriage and the defects? Yes, no, let me know, please. Do you understand why those two things are tracking together? All right, Justin says yes. Everybody is okay?
No, sir.
All right, Daniel says no. All right. When you have a small population, okay, and you're only breeding amongst this very small population, what you're doing is you're amplifying the number of rare genes in the population, okay? So let us say, for example, there's a lethal gene in a population, and it is 1 in every 10,000 people have this lethal gene in the whole population, right? So 1 in 10,000 for the entire population will have this lethal gene. But your subgroup of the population has a 1 in 1,000 incidence of the same lethal allele, okay? If you are a Pakistani from the UK and you're only reproducing with your cousins, and cousin marriages are extremely common in Pakistan. And when I say extremely, I mean it's the norm for them. It means now within that population of Pakistanis, you are breeding amongst the 1 in 1,000 chance. So the probability that two people with the lethal allele will come together and have a child that will die from the lethal allele is 10 times more than if they were breeding in the wider population, right? So therefore, breeding within small groups and keeping breeding within small groups will amplify the existence of negative genes and therefore can lead to problems, okay? And let's not just pick on the Pakistanis because Tay-Sachs disease is, remember, a disease of European Jews. And just like when the Pakistanis go to another country, they form enclaves and tend to breed amongst themselves. The European Jews do the same thing. Wherever they go, they form enclaves and they breed amongst themselves because according to each one of these groups, they're better than everybody else, so they only want to breed amongst themselves. What am I to judge, right? That said, you can also amplify potentially good traits as well. For example, the same European Jews that are susceptible to several different types of genetic disorder also have an average IQ that is much higher than the European average, okay? So I can say to you that European Jews have produced more Nobel Prize laureates than any population on the planet. And they're a small population, but they have produced more scientists, scholars than almost any population on the planet, right? Because they breed for intelligence, right? But because they're once again breeding amongst themselves for intelligence, in addition to amplifying their IQ, they also amplify the negatives of their genetics, right? You can let me know if that still doesn't make any sense to you, right? So generally speaking, nature does not like inbreeding, right? Most animals have an aversion to inbreeding. They try to prevent it as much as possible. For example, I can give you an example of elephants because elephants live in groups, right? When a male or a bull elephant comes of age, he's kicked out of the group. So the male bull elephants can't stay with the females because he would be related to all the females there. And the last thing you want is for him to be mating with those females. So they kick him out. He has to go find another group somewhere else, right? And this helps prevent inbreeding. Even dogs, everybody say, boy, yeah, dog, this dog, that, as an insult. But if you leave dogs to their own devices, brother and sister dogs tend not to mate with each other, assuming they have options for partners. Obviously, if you let them take it up in the yard, there's nothing else for them to do. So they'll do whatever. But out in the wild, they don't reproduce together. Okay? Because once again, inbreeding concentrates negative genes within a population, and it can lead to the downfall of the population, right? So cousin in Jamaica, we have a saying that cousin boil good soup. No, they don't, right? And as another thing we say in Jamaica, was sweet now, sour later, right? So yeah, inbreeding, bad. Okay. And here are some examples of some dominant inherited disorders, right? Achondroplasia. Achondroplasia is a type of dwarfism that causes the dwarfs. All right. If you've ever first, okay, I really shouldn't be using the term dwarf, since apparently it's now considered to be an insult. Maybe you've heard of this. You're not supposed to call them dwarfs anymore. You're supposed to call them little people, which, if you ask me, sounds ridiculous. But who am I to judge, right? If you're an achondroplegic little person, your disease affects the way the cartilage in your body grows, right? The chondro is for cartilage, remember, right? So in an achondroplagic dwarf, the cartilage grows very slowly. And when you look at these achondroplagic sufferers, they're the ones that tend to have the big looking heads and the short arms, short legs. Okay? Let me show you what I'm talking about here. All right. You're looking at a family here, right? This is their daughter. That is their son. Right? Daughter, son, father, mother. Right? That looks crazy. But because achondroplasia is a dominant condition, it means that both of these parents are heterozygote for achondroplasia. And since they're heterozygous, they can pass on 2 genes of achondroplasia, or 1 gene, I should say, to their daughter, and she has that type of dwarfism, or they can pass on 2 normal genes to their son, and their son grows to perfectly normal height. There was this program on TLC. Is TLC still around? I don't even know. I think it was like Little People, Big World, or something like that. I don't really watch this stuff. But if you've ever watched the show about dwarfs on TLC, you'll notice that like a family of dwarfs, pretty much all of them short, and they have 1 normal son that's normal height. I think he's even tall, right? It's because they have achondroplasia, right? There are other types of dwarfism besides achondroplasia. But this one here, as it says, is a dominant kind of dwarfism, right? Okay. Now, you might be saying, but if it's a dominant condition, shouldn't there be a lot of achondroplagic dwarfs running around the place? Guess what? Achondroplasia is one of the most common forms of dwarfism, if not the most common form of dwarfism, okay? But you also have to remember that, generally speaking, your average person is not going to want to have a child with somebody with obvious dwarfism. I mean, I think that's a reasonable statement to make, right? How many of you in here would happily marry a dwarf? A little person, I can't keep saying dwarf, right? Statistically speaking, the males in here are more likely to want to marry her than the females are likely to want to marry him. Because females in general expect their males to be taller than them, right? Males, let's be honest, there are lots of males that'll take anything, okay? But even so, statistically speaking, therefore, you're going to have fewer people, normal height people, therefore, mating with achondroplagic dwarfs. Therefore, the population of dwarfs is going to remain relatively low in comparison to the wider population, even though their gene is dominant. So you can have a dominant gene which has unfavorable characteristics. So even though it's dominant, it doesn't spread rapidly through the population, okay? Does that make sense to everybody? I'm not just talking into the
Yes,
ether,
sir.
right? Right. So being dominant doesn't mean you're successful necessarily, right? It just means that your gene is dominant. It's just like blood. The most common blood type in the world is O positive. O is a recessive gene, right? Okay, then now we have Huntington's disease. Huntington's disease is another disease that is caused by a lethal allele. So as it says, it's a lethal neurodegenerative disease. And Huntington's disease tends to kill you in your 30s to 40s, right? So it tends to kill you in the 30s and 40s. You generally speaking, have no signs, no symptoms before then all of a sudden, things start going bad for you. And then you die within I think it's 2 to 5 years you're dead. Right? Now, this is another one of these things they say, okay, if it's a dominant disease and you only therefore need one allele for this to kill you, why aren't more people dying of Huntington's disease? Because it's actually a fairly new allele. And it's normally found in Caucasians. Okay? So you have a small group of Caucasians that carry this allele for Huntington's disease. I mean, if it had shown up 100,000 years ago, it might be very common in white people. And you might have had it to the point where it significantly shortens the lives of your average white person, right? Who knows? If things continue, who knows what 1,000 years from now will look like, right? But of course, I doubt it. All right. Now, it mentions also multifactorial disorders, okay? Heart disease, diabetes, cancer, alcoholism, schizophrenia, manic depression. What am I talking about here? These diseases, right? Now, some of them you'll say, but these are lifestyle diseases. Diabetes, you eat yourself into diabetes. You eat yourself into heart disease. You drink yourself into alcoholism, right? What does that have to do with genetics? Certain genes make you more liable to become an alcoholic. Did y'all know this? I don't know if y'all know this, right? But we can actually show via pedigree chart that people who are descended from alcoholics are more likely to be alcoholics. So if your mother was an alcoholic, your father was an alcoholic, there's a greater chance that you yourself will become an alcoholic. Right? It doesn't have to happen, of course. But if you know within your family group, there's a tendency towards extremes of behavior and addiction, you have to be more careful, right? Diabetes, addiction to sugar is a real thing. As a matter of fact, there are some studies that indicate that some people, once again, this is potentially based on genetics, they become addicted to sugar and their addiction is greater than that of a cocaine addict to cocaine. So if you're that addicted to sugar because of your genetics, somebody was it Josh indicates apply it to bad temper. All right, a certain percentage of your personality is genetic. Your baseline personality, let's call it, before experiences of life and everything alter it, is genetic. Some people are genetically more aggressive than other people, right? For example, and this is a controversial example, please keep that in mind. Your average Haitian, so people from Haiti, are on average one of the most aggressive demographics, in this part of the world, right? So if you have a group of people, chances are the Haitian in there will be the most violent one, which sounds terrible when you say it out loud, right? But what can you do, right? It is what it is. That's not to say that that environment you grow up in and your own experiences can't temper a person's personality, right? Just because you have a baseline for aggression doesn't mean that you have to be aggressive. Go ahead, Daniel.
Does that mean that a lot of racial stereotypes are true?
Yeah, but Daniel, most stereotypes are based in some form of reality, right? Let's be honest. It wouldn't be a stereotype if there wasn't something to it. I mean, stereotypes are normally overblown, or exaggerated, but they're normally based on something. For example, I was listening to a program last night, and see this, and once again, this sounds terrible when you say it out loud, but here's the thing. They did a study. This was FBI released 2023 crime statistics, okay? If you're white, you have a 1 in 25,000 chance of being a murderer. Okay? 1 in 25,000. So if you pick a random white person from America, you want to have a 1 in 25,000 chance of getting a murderer. Black population is 1 in 7,870 something. That's a 79. Right? Oh, sorry, no, I made a mistake. My apologies, I made a mistake here. It's not 7,000. It's 700. So you have a 1 in 787 chance of any random black person in America you're pulling up being a murderer, and a 1 in 25,000 chance of a white person. And it's almost 1 in 60,000 if you're Asian. Right? So if you're Asian, the chance of you finding a murderer who is Asian is exceedingly low. If you happen to be black, it's not that low. Right?
How did they do this study?
Keep in mind, all right, first of all, keep in mind this study has to be based on people who are caught and convicted of murder. Right? So they catch you, they convict you of murder, and they tally up the number of murderers per year, and then they compare it to the population. All they have to do is divide. Right? So if you have a population of, I don't know, 200 million white people, and 400 white people cause a murder that year, you divide the 200 million by 400, that gives you your 1 in whatever chance. Right? I do the same thing for the black population. So that's how they came by the number. And can we also indicate that, let's be perfectly honest, black people can be very aggressive. And that is not necessarily a bad thing either, right? Because remember, aggression can be an evolutionary trait that leads to survival. If you're in an environment with a lot of aggressive, violent people, chance of you surviving increases. You yourself are aggressive and violent. It's as simple as that, right? If you're the peaceful one and everybody else want war, you're not going to make it. So there's evolution involved, okay? Schizophrenia. Schizophrenia, which of course is a mental condition. And I know normally people hear schizophrenia, they think about people with multiple personality disorder, but multiple personality disorder is just one type of schizophrenia, right? You have people who talk to themselves, people who, you have those who twitch and scream. You have people who think they're being watched all the time, all of these will be considered schizophrenics, right? And once again, there is a link, there's a genetic link. The more people you have in your family tree, in your pedigree chart, that would have been diagnosed schizophrenic, the greater the chance that you will be diagnosed as a schizophrenic as well. But also remember, genetics has to play in the environment, right? So you can have somebody who has a propensity towards schizophrenia, but they have a nice, quiet life. All is well around them. People around them love them and treat them good, and all is well. And they go through life. They never had have an outbreak. Never have a breakdown. They have their children. Something goes wrong in the children's lives. The children get stressed, and all of a sudden, boom, schizophrenia. Right? So having the gene for something doesn't necessarily mean you must get it. It's like manic depression. Do you know what manic depression is, by the way? I'm assuming you all have an idea as to what depression is. But do you know what manic depression is?
Oh, sorry.
People who have this manic depression are people who suffer from extreme mood swings. Outbursts, sort of. So a manic depressive can be somebody who's extremely happy and, and then the slightest thing turn them completely in the opposite direction. So they go so bipolar is a offshoot of manic depression. So yeah. So if you know somebody with bipolar disorder, it falls under that. So as you said, their mood can swing from one extreme to the next. They can be very, very calm 1 minute and screaming and throwing things the next. or they can be very, very happy 1 minute and suicidal the next. Right? Once again, if it's in their family, it might show up in them. Which is why my advice is this. If in the future you decide you want to get married, meet the person's parents, meet their grandparents, meet them uncles and them auntie, and see if those people are normal. If there's an uncle or something that they don't want you to meet, assume that person is not normal, and the more of them you don't want to meet, you just kind of de-rank the person in terms of whether or not you want to get involved with them, right? If they're like, no, we don't really need to meet my mother, what wrong with your mother? No, my uncle, him dead about, you don't have to worry about him. What wrong with the uncle? Right? So you're doing your own pedigree chart, basically, of craziness. Because remember, do you want to add your genes to the crazy tree? You have to think about these things, right? Anyway, moving on. We're not going to worry so much about genetic screening. I'm just going to give it a basic idea. These days, a simple DNA test is fairly easy to do. Let's say, for example, you're at risk of sickle cell, since of course, Caribbean, we deal with sickle cell. You're at risk of sickle cell disease. You have the sickle cell trait, your partner has a sickle cell trait, the baby is coming, all right? The pregnancy is there. You want to know if you're going to give, if your child is going to have sickle cell disease. You can do an amniocentesis fairly early in the pregnancy, okay? You can do the amniocentesis within 6 weeks of the pregnancy. What they'll do is that they'll essentially stick a needle into the abdomen, right into the amniotic sac, and pull out, as it says here, about 10 mils of amniotic fluid. Why are they doing that? Because floating in the amniotic fluid, you'll have skin cells from the fetus, and you'll also have placental cells, right? And the placenta also comes from the fetus. So you can then do a DNA test on these cells to determine if the alleles you're worrying about are showing up in the offspring, right? Now, has been a lot. Keep in mind, this is not super common yet, but some European countries are going to make it the law that all children have to have a DNA screening at birth. Okay, I think it's a good idea. Because these days, DNA screening is getting cheaper and cheaper and cheaper, right? So this is showing you how the amniocentesis is done. So if you have a child, or if you do the DNA screening before, and you know the child is going to have serious, I shouldn't say child, it's just a fetus if it's not born yet, right? And the fetus is going to have serious problems. Most countries with abortion will allow for an abortion of a fetus that has obvious genetic problems, okay? Maybe you don't want to have a child with Down syndrome. Maybe you don't think it's a blessing, which of course it's not. Maybe you don't want to have a child with severe mental retardation, right? Or a child that is going to have sickle cell disease that will suffer for years and years and years before it eventually dies. Then you can have that child aborted. And by doing this, you're doing multiple things. One, you're reducing the population, which is always nice. And two, you're reducing the number of bad genes entering the population into the next generation, okay? Right now, we're having a problem. You probably don't even realize this. But in the last 100 years, medicine has gotten so good that lots of people who should have died are around having children, right? People with significant genetic disorders that should have died before they reproduced are reproducing. And which sounds great, except for the fact that the percentage of people with bad genes in your population is starting to increase, which means that you're degrading the genome of your species by allowing this, right? Evolution is very harsh. It's a harsh scrubbing. So, so evolution is constantly scrubbing your population to get rid of the worst of it. But we are stopping the scrubbing from happening. For example, hemophilia is more common now than it was 100 years ago. Because hemophiliacs are being treated. We have very good treatments for hemophilia. So hemophiliacs are happily having children, right? And passing on the hemophiliac gene to these children who are potentially hemophiliacs themselves or will have children who are hemophiliacs. So there are now more people dependent on the pharmaceutical companies to stay alive because of it, right? I can say the same thing with in Jamaica for sickle cell disease. Normally, you're born with full-blown sickle cell, you don't make it at the age of 12. I mean, I know men who have children, they have full-blown sickle cell. They pass on a trait to their children. Women, there are some women who have full-blown sickle cell, and women with full-blown sickle cell, the doctors tell you no, do not have children, it can kill you. And they still go ahead and have children, and some of them make it, some of them don't, right? So which means that they're still passing on the sickle cell trait to the next generation, which means, of course, you're going to have an increase, right, over time in the prevalence of these things in the population, if that makes sense to you, right? So yeah, it sounds harsh, but you know, nature normally would not allow this to happen, but mankind does, okay? So having mandatory genetic testing might be a very good thing, because at least the person knows from day one, I am susceptible to XYZ. When they have their partner, they can say, partner, this is what I am susceptible to. What are you susceptible to? And if the two of them have something that is too bad together, go find somebody else, right? You can make it first date business. The first time I go out, you hand her your docket, you give her, she hands you her docket, and then sit down and go to the docket and say, yeah, this is not going to work out, you know. And then go on a yard, you know? All right, moving on. So, chi-square test. You all said you haven't done chi-square test yet, okay? So let us quickly go through, well, let's go through chi-square test to make sure we're all on the same page here, okay? The chi-square test is going to be one of the first tests you will learn how to do in statistics, okay? As I said, it's a statistical test, including chi-square, I used to determine and to test for significance of experimental results. The idea here is that you set up an experiment, you have now received a result. You want to know if the result that you have received is so different from what the norm would be that your results are valid, okay? You want to validate your results. So you can use a chi-square test to do this. So as it mentions here, the chi-square analysis is performed to test if the observed results are close enough to the expected results that any difference between observed and expected is probably due to chance. So for example, right, if I want to test a drug, let's say, okay, I want to test a drug that is supposed to prevent flu. What the hell? Let's use corona. What the hell? Since they actually have drugs for this. I want a drug that is not even a drug. Let's use the same vaccine. So here we have a corona vaccine. It was supposed to prevent you from getting corona. What do? You get two cohorts of people, or you get one massive cohort, let's say 5,000 people. You give all 5,000 people the vaccine, right? Then you have another cohort of 5,000 people, and you don't give them the vaccine. Now, you follow them, let's say, over a period of 6 months. At the end of the 6 months, you tally up the number of people who got corona in each group, right? Let's say 10% of the non-vaccinated group got corona, and 6% of the vaccinated group got corona. You want to know if the difference between the 10 and the 6% is significant. Why? Because you want to be able to claim that, hey, my drug prevents you from getting corona. So you do a chi-square test comparing the results to see if your drug, right, the results of giving the person your drug is better statistically than a person not getting your drug. And the chi-square test will be used as proof of that. Okay? So that's one of the reasons you'll do something like a chi-square test. You want to know if what you expect to get is different from what you're observing. Okay? You want to know if what you're hoping for and what you got are close or far apart. That's the basic idea, right? So, chi-square tests require two types of hypotheses, right? We have what is called a null hypothesis. That's not how you write null. All right, let me show you how to write null. It's just that it's hard to find a symbol in Word. This is null. Okay? So that's the null hypothesis. Of course, we type it this way because that's easier. You also have what is called an alternative hypothesis, which is written like this. Okay? So we have null hypothesis, and we have an alternative hypothesis. The null hypothesis is a statement, right, that indicates that when you've done your experiment, the results you get and the results you expect to get, there's no difference between them. Right? Remember, if I'm saying anything that doesn't sound sensible, let me know, right? So your null hypothesis will state what you expect to get and what you get are close enough that there is no real difference between them. Okay? So if I did the same corona experiment we mentioned before, my null hypothesis would be that my vaccine does not reduce the risk of getting the coronavirus. Okay? Now, why would I want a null hypothesis? Very simple. You want something to disprove. You want your results to disprove what you have. So therefore, the null hypothesis is normally written in a negative way. So there is no difference. This thing doesn't work. That's what you want to disprove. So you want to disprove your null hypothesis. Okay? So far, so good. No questions so far, right? Then now your alternative hypothesis is the one that you want to prove. You want to be able to say, the null hypothesis is wrong. My vaccine does work. The vaccine reduces the chance of catching corona, right? And your chi-square test will allow you to indicate either your null hypothesis is correct or your alternative hypothesis is correct. Okay, that's the purpose of the chi-square test. All right, so far so good, right? Let's do a simple example of a chi-square test. If you toss a coin 10 times, right? Assume all things being equal, the breeze is blowing at the same rate, the local gravity remains constant, everything you can think of remains the same. You expect to get an equal number of heads and tails, right? Flip a coin 10 times, you're supposed to get 5 heads, 5 tails, assuming that their outcome is random. If you have random outcome and, well, 2 outcomes that are possible, and it's random, you're supposed to get equal possibilities, right? Now, as it mentions, what if you got 8 heads and 2 tails? Does that mean something's gone wrong? Does it mean that the coin you're flipping is a bad coin? Somebody's trying to play with you, right? Or not? So a weighted coin is one and it's slightly heavier on one side, and the side that is heavier on tends to be the side that goes face down. So for example, you have some people who like to trick people with coins, so they'll get a weighted coin and they'll say, I bet you I can flip this coin 10 times and I'll get 8 heads. If I win, you give me $100. If I lose, I give you $100. And of course, that your $100 gone, right? So it's a trick. It's the same thing with dice. Sometimes, people play dice for money, and they'll use their own dice, and the dice are weighted. So you play sevens. So when you roll the dice, you get 7 more often than not. Right? That's why, for example, when you go to a casino, you can't bring your own dice home, you have to use a casino dice because of that, right? Anyway. So let's translate that now into this. This is the equation for chi-square. All right? So the chi-square value here, unless the chi-square should be written like this. But once again, it's hard to do this in Word. What you have here is the sum. Remember, this symbol here means sum, right? Of the observed results minus the expected results squared divided by the expected results. And this will give you a chi-square value that you can use to determine whether your null hypothesis is correct or your alternative hypothesis is correct, okay? This means, of course, that we need to have observed, and we need to have expected, okay? In the example we gave earlier, when the coin flipped 10 times, we expect to get 5 heads, 5 tails, assuming it's a normal coin, okay? But when they did the experiment, they got 8 heads, 2 tails. So what we need to know now is if 8 and 2 is close enough to 5 and 5, then they can say, yeah, the coin is normal. Or if it's so different that you have to accept the alternative hypothesis that there's something wrong with the coin, okay? So here we have observed and expected. So what we do is we plug these into our equation. So first thing so this is our equation, remember, right? So we have observed minus expected squared. So you have observed, which is 8, minus expected, which is 5. Right? Remember, 8 minus 5 right here. Then you have 2 minus 5 here, right? So 8 minus 5, 2 minus 5, okay? So that's how we came by this. So observed minus expected squared. Now remember, when you square a number, and this doesn't make a lot of sense to me, but I'm not a math person, multiplying 2 negatives gives you a positive, which sounds completely ridiculous, but hey. That now gives you this. We have 3, so 8 minus 5 gives you 3, and 2 minus 5 gives you minus 3, okay? You then square the numbers to get whole positive numbers, which of course means 3 squared, of course, gives you 9, okay? So now we have 9 plus 9, okay? Divided, of course, by the expected down at the bottom, which is the 5, okay? So that should be under here. That should be under there, right? So that now is, so 9 divided by 5 gives you 1.8, okay? And of course, this one says sum. So when you sum that together, you now get a number that is 3.6. Okay? No questions so far, so I'm assuming all is well. Then now, once you get this 3.6, this is your chi-square value. In order to determine whether it's the alternative hypothesis or the null hypothesis you use, you are then going to need a table of critical values. All right? Like this one here. This is a section of a table of critical values. So once you get the chi-square number, you need to get the chi-square statistic from this chart, okay? Here's how you use the chart. You need a couple of pieces of information to use this chart. First one you need to know is what we call the degrees of freedom, okay? The degrees of freedom is, you get it by using a formula that is n minus 1, okay? N minus 1 is the number of categories. So number of categories of data we have minus 1, okay? And for us, we have 2. We have, you can either have heads for the coin and tails, right? And we minus 1 from that. So we end up now, of course, with 2 minus 1. So we have 2 categories, heads and tails. So that's 1 plus 1, which gives us 2 minus 1, okay? So that is degrees, or degrees of freedom equals 1, okay? Once we have our degrees of freedom, we then need to look on the table. So remember, our degrees of freedom, the DF, equals 1. So when you look at the table now, we have columns marked DF. Our D equals 1, so we pick column number 1. Okay? We then need to know what row to look in. And the row we use is based now on the probability level. Unless you are told otherwise, always use 0.05. Let me repeat. Unless you are told otherwise, you will always use 0.05. All you gotta do is just like the Punnett square, is you look at the number that coincides with column and row, which gives me 3.84. 3.84 is now my chi-square table value, right? Or critical value. The calculated chi-square value was 3.6. The table value is 3.84. All you have to do now is you compare these two numbers, okay? As long as the chi-square value is less than the critical value, you accept the null hypothesis, right? In this case here, it means that there is no statistical difference between observed and expected. So you accept the null hypothesis for this. 3.6 is less than 3.84. Therefore, we accept null. All right. Any questions so far? Okay. So let's use a Mendelian example now, okay? We have a pea plant here. We have a trait, this is T for tall. All right, we have 2 heterozygote plants, okay? Then we cross these 2 heterozygote plants. We get 520 seeds from the plant. We plant the seeds and allow them to grow. And when we grow the plants, we end up with 338 tall ones and 182 short ones. So this, of course, is our observed, right? So we see this. We want to know if this observed is what we are expecting to get, right? Remember now, this is Mendelian genetics. So if you do a quick table, and you know how this go already, we end up with this. Okay? So end up with 3 tall to 1 short. Okay? So 3 to 1 ratio. So we're expecting 3 times more tall plants than short plants. Okay? We have a total number of plants of 520. So we need this 520 in this ratio of 3 to 1. So here's what we do. We add 3 plus the 1 together, okay? So 3 to 1 equals 3 plus 1, which equals 4 parts. All right? Then now all we gotta do is divide the 520 by 4. this gives us 1 part. So that now means, of course, that in our 3-to-1 ratio, the 1 is actually 130 or 120? 130, right? So that's 130. And the 3, of course, is 3 times 130, so that's 390. So this is what we expect to get, okay? So we can do a table like this. So we have our observed and expected, right? So we have tall plants. What did we observe? 338 to 182. So 182, which gives us our 520. Expected is 390 to 130. It goes the same 520, right? Then, of course, we have observed minus expected. So that's 338 minus 390. That's minus 52. And then, of course, the next one is 180 minus 130, which gives us 50. Okay? So that's observed minus expected. Wait, how do I get 52? Did I add up something wrong? Does this look right to everybody else? Did I miss something here?
So it's 182 and you have 180. Well,
Oh.
182 minus
Oh, okay, I
130.
see. Yes, that makes sense. That's 182. So that should be 52. Okay, so that makes sense. All right, thanks. Cool. So we have positive 52, negative 52, right? So of course, we then do our observed minus expected squared. So when we square 52, we get 2,704, nice big number, okay? We then divide by the expected. So the expected for the tall plants is 390, and for the short plants is 130. So we end up now with 6.93 and 20.8. We sum them together, so add them, now we get 27.7, okay? Now remember now, we need n minus 1. Once again, we have 2 categories. We have tall plants, short plants, so that's 2 minus 1. So once again, our degree of freedom is 1. So when you go to the chi-square table, of course, we get our value of 3.84 again. And since our calculated chi-square is 27.7, that's way more than 3.84. Therefore, there is a difference between what we observe and what we expect. So the way this plant, we go back to the plant here, 338, 182, the way height is being passed on from one generation to the next generation is not Mendelian, right? Which of course means that we can formulate our null hypothesis in this way. We can state that the plants are passing on their traits in a Mendelian way. And in the alternative hypothesis, the plants are not passing on their traits in a Mendelian way. And we can conclude, based on the results here, that the alternative hypothesis, which is the plants are not passing on their genes in a Mendelian way, is correct. Okay? So for the first one we did with the coin flip, we accepted the null hypothesis as being true. In this one here, we reject the null hypothesis and have to accept the alternative hypothesis. All right. The rest of this here is just more information. And it's pretty much the same information on the chi-square that I just mentioned. So please read through it. It gives an example here in a dihybrid cross. All right. So let's run through the dihybrid cross example, okay, real quick. So we have here two traits we're looking at. We're looking at black bodies, and we're looking at eyelessness. Okay? Or blacks, sorry, what I should say is bodies and eyes. So we're looking at bodies and eyes as the traits. Okay? The body can be black, the body can be the wild type, which is orange. You can have eyes or you can have no eyes. Okay? Those are the traits we're looking at, two traits. So they did a cross between a homozygous dominant over here and a heterozygous dominant. Sorry, homozygous recessive to give us a heterozygous dominant, right? So let's see what they used. So if we were doing genotype thing, we could say the body is either the wild type or it's a black. So we use W for wild type. So common W versus capital W for the color. And then for the eyes, it's either eyes or no eyes. So I'll say capital for eyes, common for no eyes. Okay. So the offspring here is going to be, of course, heterozygous for both traits. And then now, when you cross heterozygous offspring, this is what you get. Okay? You get 5, 6, 10 of the normal wild type, which of course means orange and the eyes. You get 1881 orange with no eyes. I'll just put N for no, right? And then you get 1896 black with no eyes. And 622 black with eyes. Right? So we have 4 categories this time instead of 2. So chi-square test for this. Now, in normal Mendelian genetics, when you do a dihybrid cross with two, of course, genes, you're expecting a 9:3:1 ratio, right? So the question is now, are these numbers indicative of a 9:3:1 ratio? So that's what we're going to do now. So here we have the table. We have a total number of 10,009, right? But a 9:3:1 ratio gives us 16 parts. So far, so good. So just like we had before, we add up the ratio to get the number of parts. If we got 16 parts, here's how we get our expected. It shows you here. It says 916 times 10,009. So 9 divided by 16, of course, gives us 0.562 times 10,009. And I get here 5,630. How do I get 5,634? I have no idea. But let's work with what they have. And the 3 divided by 16, they must be rounding off somewhere. They probably round that off to 19. Let's see. Multiply by 10,009. They get 1878. We got 1876. Yeah, man, they're rounding off. So 3 divided by 16 equals that. So if we round off to 0.19, let's see how much we get. Yeah, I have no idea how they're rounding these numbers off, but let's work with their numbers, okay? So you see how I got the numbers, right? So 9/16. So 9 divided by 16 times the total, 3/16. 3 divided by 16 times the total, right? So once they've done working that out, these are the numbers they would have got, okay? So we have observed and we have expected. So we can plot down observed and expected in the same table. So expected 5, 6, 3, 4, 1878 twice, and 626. So here we have it. This says hypothesis for some reason. Should say expected. Okay. So we got our observed, we got our expected. So of course now we run through the chi-square formula. We have observed minus expected squared divided by expected. And then that's how we come by the 0.07. Let's make sure. You know the numbers have been a bit funny. So we have 5610 minus 5630 gives us minus 20. You square that. That's 400. Then you divide the 400 by 5630 and you get 0.07. So that's correct, right? So we can assume the rest of them are correct as well. So we have 0.07, 0.01, 0.2. So that gives us 2.8. And then we have another one here, 0.02, which adds up to, of course, 0.3. Okay? Now, remember, we have 4 categories. So n minus 1 is therefore 4 minus 1, which gives us a 3. So 3 degrees of freedom. So when we go to the table now, remember now, we have 3 degrees of freedom. So we start here. And remember, as long as you're not told otherwise, use the 0.05 column, which is here. Right? Then now we look where they meet. Which is 7.82. All right. So remember now, we have 0.3 and we have 7.82. Obviously, 7.82 is much higher than 0.3. Right? Therefore, we accept the null hypothesis that the numbers that we have here are close enough to the 9:3:1 ratio that we can accept this is Mendelian dihybrid cross. Okay. Any questions or are we good?
That's a good question. So
Yes.
if the number is lower than the expected value is
Not
null,
the
and if it's
expected
great,
value, than the table value.
the table value
So
is
the table value
that
is 7.82.
it's null, and if it's greater than it is alternate.
Alternative, yes.
So what's when it's equal? Will it be null as well if it's equal
Generally
to it?
speaking, if you see them and it's exactly the same, you assume it's an alternative because it means that it is so close that chances are something is going on. So you err on the side of caution, which is that something is going on. All right, any other questions?
Sir, what if it's really close but not yet there?
In what direction, above or below?
Below.
As I said to you, if it is the same exact number, then you will assume that there is a significant difference. Sorry, no, I made a mistake. You assume the null hypothesis. My apologies. So if it is pretty much the same number, you assume that nothing is going on. There's no difference. You accept the null hypothesis, right? Because you're trying to prove a difference. it should be harder to prove a difference than to prove no difference. Therefore, if it's close, if it's extremely close, more likely than not, you have to say that there's no difference, right?
Okay.
But usually you don't have to worry about the numbers being that close, right? Remember, we're going down to 2 decimal points. So usually you don't have an issue. All right, so hopefully if you all see a chi-square question, you can do a chi-square if asked to do so, okay? All right, so we stop here for today. Tomorrow, we will start on mutations, okay? So I'll see you all tomorrow at 8 o'clock. And I think I'm seeing you later today at 11. It's 11 o'clock, right? Yeah, I'll see you in an hour. All right, folks, anatomy people, I'll see you all in an hour. Otherwise, I'll see you all the rest of you tomorrow.